Welcome back, everyone. This is part 2 of the 1.3 mini lecture on macromolecules, and we're going to pick up part 2 with the discussion of proteins. So proteins are the predominant organic molecule in cells, and proteins are responsible for pretty much every physical trait that you can observe in yourself and other organisms. They are composed of 20 different amino acids, and we distinguish proteins by their size. So a peptide is a molecule composed of short chains of amino acids, typically less than 20. A polypeptide usually has more than 20 amino acids and is generally a smaller subunit of a larger protein. And then we consider a protein any structure that contains 50 or more amino acids. Proteins also are diverse in structure in how their amino acids bond and form additional, more complex structures. So when we're talking about a protein's primary structure, we're talking about the type of amino acids, the number of amino acids within that change, and then the order of amino acids. So we're talking about just the amino acid chain itself. The secondary structure refers to the shape that this chain then takes. And this shape forms when the functional groups within the amino acids interact with each other and form hydrogen bonds. And a couple of different shapes can be produced. We can either produce an alpha helix or a coiled structure, or what we call a beta pleated sheet. And we'll look at examples of this on the next slide. Tertiary structure forms when additional bonds are formed between other groups within the amino acids. And this tertiary structure leads to a very complicated folding of that protein chain. And it leads to the functionability of the protein itself. Finally, we have quaternary structure, and this structure does not occur in all proteins. When we're talking about quaternary structure, we're talking about more than one polypeptide folding together to form a larger multi-protein subunit. So here is our primary and secondary structure, and you can see primary structure, we're just talking about the chain itself of amino acids and what are those amino acids, how many are there, what's the order, okay? The secondary structure then forms as the functional groups in these amino acids form hydrogen bonds, and those hydrogen bonds lead to additional sort of folding and shaping of the protein. This is a beta pleated sheet, and this is the alpha helix. So depending on how these amino acid groups form those hydrogen bonds will then determine what shape the protein takes in its secondary structure. And then finally, we have the tertiary and quaternary structure. And when we're talking about tertiary structure, we're talking about additional folding and bending that creates sort of these active sites on the protein where the protein then becomes functional. It's able to do whatever its job is. Quaternary structure, then we're talking about two or more polypeptide chains that have folded together to create a functional larger unit. Every protein develops its own unique shape, and this is how we distinguish proteins, or one way that we distinguish proteins. And on the surface of that folded structure, then we have these very distinct pockets or bulges on the protein, and those pockets and bulges then become active sites for other molecules to interact with the protein, similar to a lock and key. This is how enzymes work. The enzyme is the protein, and it has particular features on its surface that other molecules are going to interact with, and that's how it helps to catalyze or speed up particular chemical reactions within cells. Antibodies also work in this way. This is how glycoproteins with specific regions for attachment are going to interact with the antibody to identify bacteria, viruses, or other microorganisms. So we can find proteins in two different states. The native state is the functional state, and you can see here it's folded normally. You can see it's got a functional active site. Okay, this is the normal or native form of the protein. When a protein is denatured, the native state then becomes disrupted. The protein begins to unfold. The active site is no longer present, and this can happen in a number of ways. First, heat. Heat is probably one of the primary ways that proteins are denatured. This is why we cook our food, because the proteins denature and become more pliable and easier for us to break down. Proteins can also be denatured by acid, alcohol, and some disinfectants. Okay, so when a protein is denatured, it is unable to perform its job as it normally does. All right, so now we're going to talk about our last group, which are the nucleic acids. And nucleic acids contain the instructions for all of the functions and structures within our body. DNA is one form of nucleic acid, and DNA, or deoxyribonucleic acid, contains the genetic information which provides detailed and specific instructions for our heredity, for our traits. RNA, or ribonucleic acid, is the molecule that is responsible for reading that DNA message and then carrying that translated message from the nucleus out into the cytoplasm so that proteins can be formed based on those instructions. We have also discovered that small RNA fragments are also key in DNA regulation. So let's look at DNA. Most of you are probably familiar with this double helix structure of DNA. We call it a double helix because we have two helices, or two, how to describe this, cyclic patterns that are bonded together. And there are 4 nitrogenous bases that form the structure of DNA. And you can see them here: adenine, guanine, thymine, and cytosine. And they have different structures. So thymine and cytosine are single carbon ring structures, and they are part of a group called the pyrimidines. Adenine and guanine have double ring structures, and they are part of a group called the purines. Adenine always bonds with thymine in the structure of DNA, and guanine always bonds with cytosine in the structure of DNA. And when they bond, they form hydrogen bonds between them. Okay? So if we look here, here's adenine, here's thymine, and between these two molecules, then, this is where the hydrogen bonds form that join those two helices, or those two ladders of the DNA molecule. Okay. The nitrogenous bases, or I should say the nucleotides themselves, are composed of 3 parts. There is a phosphate group, a 5-carbon sugar, and a nitrogenous base. Okay, and we talked about the nitrogenous bases. In DNA, they are adenine, guanine, thymine, and cytosine. And you can see here, purines are adenine and guanine, pyrimidines are thymine and cytosine. And the 5-carbon sugar in DNA is deoxyribose. In RNA, RNA is a single-stranded molecule, not double-stranded like DNA. And there are a couple of things that are different between RNA and DNA. First, RNA is composed of ribose. So instead of deoxyribose, RNA is composed of ribose. It is also a 5-carbon sugar. There is a phosphate group that is attached to this 5-carbon sugar, and then we have the nitrogenous base. However, in RNA, instead of thymine, we have a molecule called uracil. And in RNA, adenine and uracil are always going to bond to each other. Okay. So RNA, as I said before, is single-stranded, and there are 3 different types of RNA that we can find within cells. The first type is mRNA, or messenger RNA. Messenger RNA is produced within the nucleus of eukaryotic cells. And messenger RNA is going to read the message that's contained, read the instructions that's contained in the DNA molecule, and then it's going to carry that message from the DNA to ribosomes so that proteins can be made. tRNA, or transfer RNA, is a carrier protein, or not a carrier protein, a carrier molecule, that is going to deliver the correct amino acid that matches a 3-code message, and it's going to carry that amino acid from the cytoplasm to the ribosome so that proteins can be assembled. rRNA, or ribosomal RNA, is the component of ribosomes. So it is important structurally for the formation and function of ribosomes. Okay, and we also know that there are small fragment RNA molecules that are responsible for DNA regulation. Okay. ATP, or adenosine triphosphate, is also a nucleotide. However, ATP is not involved in protein synthesis or heredity. It does not contain genetic information. ATP is the energy molecule of all cells, and it is composed of 3 things: adenine, which is the nitrogenous base, ribose, the 5-carbon sugar, and then it contains 3 very high energy phosphate molecules. And when the bond is broken between the 3rd and 2nd phosphate, we get a lot of energy released. And this is how our cells, all cells, are able to perform their functions because of the energy contained in this molecule. And you can see here, this is ATP. So here's our ribose sugar. Here's our nitrogenous base. And then here are our 3 phosphates. And when the bond is broken between this 3rd and 2nd phosphate, then we get a huge energy release. Okay, so that's the end of part 2. Here are some questions that you should ask yourself to review to make sure that you understand the content from part 1 and part 2, and I hope this video was helpful.